respiratory system. The functions of the respiratory system are to supply the body with oxygen and to remove carbon dioxide from the body.
Here we have the upper respiratory anatomy. Air first enters the external nares, which are the nostrils. You can see it here, it doesn't show on this side. And then it enters the nasal cavity.
The functions of the nasal cavity are to house the smell receptors and to clean, warm, and moisten the air that you breathe.
In the nasal cavity, there are two types of mucosa. The first is olfactory mucosa, which contains smell receptors, and the second is respiratory mucosa, which lines the back of the cavity here. Respiratory mucosa is made of pseudostratified ciliated columnar epithelial tissue. In the nasal cavity, we have the superior, middle, and inferior nasal conchae. The floor of the nasal cavity is the hard palate and the soft palate with the uvula on the end. And here is the internal nares. Also part of the upper respiratory system are the paranasal sinuses. These are hollow areas in the bone. Here we have the frontal and the sphenoid. This model only shows 2 of the 3 paranasal sinuses. The ethmoid is not shown.
The function of the paranasal sinuses are to lighten the skull, to warm and moisten the air that you breathe, and to give your voice a tonal quality.
The pharynx includes the nasopharynx, the oropharynx, and the laryngopharynx.
Commonly known as the voice box, the larynx is shown here on this model and in more detail on this model.
The functions of the larynx. The larynx keeps the airways open, makes sure food goes down the esophagus and not the trachea, and produces voice.
When we open up this model, we can see the epiglottis right here. Inside the larynx, we have the false vocal cord and the true vocal cord. The false vocal cord functions to protect the true vocal cord, and the true vocal cord is what vibrates to form sounds.
The air travels down the trachea. This is the carina of the trachea, where it splits into the right and the left primary bronchi. Primary bronchi are the big ones here. That splits into secondary bronchi, which then splits into tertiary bronchi. The tertiary bronchi are the little ones on the end. The tertiary bronchi become terminal bronchioles. The main difference between bronchi and bronchioles is that bronchioles do not contain hyaline cartilage, where bronchi do. The terminal bronchioles become the respiratory bronchioles, as you can see here and here. Respiratory bronchioles are actually where the exchange of gases start. The alveolar ducts are passageways to the alveolar sacs. It's kind of hard to see because all the alveoli are covering, but it's here and here. The alveolar sac is a cluster of alveoli. They contain type 1 cells and type 2 cells.
Type 1 cells form the alveolus and are made up of simple squamous epithelium. Type 1 cells are also part of the respiratory membrane or air-blood barrier, which is made up of the alveoli, capillary walls, and fused basal lamina. Type 2 cells are also called septal cells. These cells produce surfactant, which acts like a soap, breaking down surface tension of water so the lungs don't collapse. Blood supply to the lungs includes both incoming deoxygenated blood from the heart and systemic circulation. Deoxygenated blood from the heart enters into the lungs through the pulmonary arteries. Pulmonary capillary networks that surround the alveoli then absorb oxygen from the blood. Pulmonary veins bring blood out of the lungs. Just as the rest of the body requires oxygenated blood, the lungs do as well. Bronchial arteries bring oxygen-rich blood to the tissue in the lungs, and then capillaries around conducting passageways of the lungs supply oxygen to these areas. Bronchial veins add a bit of deoxygenated blood to the oxygenated blood.
Pressure relationships in the thoracic cavity. Intrapulmonary pressure is pressure within the alveoli inside the lungs. Basically, that's atmospheric pressure when between phases of inspiration and expiration, not breathing in and out. Intrapleural pressure is pressure within the pleural cavity. So it's the pressure outside the lungs. Transpleural pressure is the difference between intrapulmonary pressure and intrapleural pressure.
Each of the respiratory muscles is found in the trunk of the body. The diaphragm contracts, flattens, and moves downward. The external intercostals cause inhalation, and the accessory muscles include the sternocleidomastoid and the pectoral to increase volume of the thoracic cavity.
Respiratory movements. Eupnea is quiet breathing. Diaphragmatic breathing is deep breathing. Coastal breathing is shallow breathing. And hyperpnea is forced breathing or forced expiration.
There are 4 physical factors influencing pulmonary ventilation, which includes respiratory passageway resistance, lung compliance, lung elasticity, and alveolar surface tension forces. Respiratory passageway resistance is determined by the lumen size of airways, as well as constriction or dilation of smooth muscle of bronchioles. For example, the parasympathetic nervous system is stimulated by irritants and causes constriction, which then causes increased resistance. The sympathetic nervous system causes bronchodilation and decreased resistance. Lung compliance is how easy it is to stretch the lungs. This is related to elasticity of tissues and walls in alveolar ducts and is decreased by diseases such as tuberculosis and pneumonia. As previously stated, surfactant acts like a soap and helps break down surface tension in the lungs.
Respiratory volumes. Tidal volume is how much air you breathe in and out normally. Inspiratory reserve volume is how much you can inhale after a normal inhalation. Expiratory reserve volume is how much you can breathe out after a normal exhalation. Residual volume is what's left in lungs after expiratory reserve volume. Pulmonary function tests. The spirometer measures the amount of air moving in and out of lungs. Total ventilation, or minute ventilation, is the amount of air moving in and out of your lungs per minute. Forced vital capacity is when you breathe in as much as you can and then breathe out as much as you can, measuring the outgoing gas. Forced expiratory volume is measuring gas over time. Non-respiratory air movements include coughing, sneezing, hiccuping, and yawning. Gas exchanges between the blood, lungs, and tissues. There's partial pressure gradients and gas solubilities. Then there's thickness of the respiratory membrane, which can affect pulmonary gas exchange. The surface area for gas exchange is when there's more surface area, there's more gas exchange. Ventilation-perfusion coupling is how the respiratory system are backwards according to oxygen. Internal respiration is capillary exchange in the body tissues.
About 1.5% of oxygen is carried dissolved in plasma, and about 98.5% is carried on hemoglobin. Factors that influence hemoglobin saturation include the percentage of oxygen, temperature, pH, and percentage of carbon dioxide. As the percentage of oxygen increases, hemoglobin affinity also increases. As temperature increases, affinity decreases. As pH decreases, affinity decreases. And as percentage of carbon dioxide increases, affinity decreases. About 70% of carbon dioxide is carried as bicarbonate ion in plasma, about 20% is carried as carboaminohemoglobin, and about 10% is carried dissolved in plasma. Being that carbon dioxide in blood is acidic, it lets you control blood pH with breathing.
Neural mechanisms controlling respiration. The medullary center controls the rhythm of breathing. The pons respiratory center fine-tunes breathing and prevents overinflation of lungs. Respiratory adjustments include exercise and high altitude, while clinical problems include chronic obstructive pulmonary disease, which is permanent enlargement of the alveoli. It also includes tuberculosis, which is creating resistant strains, and a lung cancer, various forms including squamous cell carcinoma, adenocarcinoma, and small cell carcinoma.